Today I will discuss the action potential of cardiac contractile cells and how this electrical signal produces coordinated pumping of the heart. I will explain the ion gradients which create the membrane potential, describe the phases of action potential, connect calcium handling to muscle contraction, and we will discuss clinical implications. This understanding will be derived from cardiac physiology research and reviews from Sedlak and colleagues from 2023 and foundational physiology texts from Sherwood 2025. Cardiac contractile cells make up most of the atrial and ventricular walls. Unlike pacemaker cells, they do not depolarize spontaneously. Instead, they rely on the conduction pathway to trigger activation. Gap junctions electrically connect the neighboring cells, which allow the heart to contract as a single coordinated unit known as the syncytium. The cells can maintain a stable resting membrane potential near -90 millivolts until stimulated, which is important for reliability and powerful contraction. This table gives a clear summarization of key ions involved in generating membrane potential. Potassium, which is the first ion listed on the table, is involved in generating the muscle membrane potential. This is going to be high inside the cell, but low on the outside of the cell, which encourages potassium to exit the cell and make a negative interior environment. Sodium, which is second on the table, is also low inside of the cell, but high outside. This allows a strong force for sodium to enter when the sodium channel is open. Moving on to calcium, third on the table, calcium shows an even greater gradient, almost zero inside of the cell, but on the outside of the cell, it is high. This is a great example of why calcium entry is very powerful in triggering contraction. Chloride remains higher on the outside of the cell, which allows stabilization within the gradient charge. These ionic differences allow for the electrochemical foundation, which shapes the cardiac action potential. Here we have the linkage of the cardiac conduction action potentials within different tissues and the ECG. At the bottom, we have what is called a timeline. The horizontal axis labeled 200 milliseconds. This means each tick represents 200 milliseconds, so the entire strip shown here covers several milliseconds of one heartbeat. This visualization allows us to see when different regions are depolarized and relative to each other. The dots on the left-hand side show the heart trace of the conduction pathway, starting at the syncytial node, known as the SAN, located high in the right atrium, shown by this arrow here. Moving to the waveform labeled SAN, right here at the top, this demonstrates a very slow but rising depolarization during phase 4, due to the pacemaker cells gradually drifting towards the threshold. The upstroke is slower than the muscle because it is calcium driven. After the depolarization, the membrane repolarizes and the cycle repeats, setting the normal heart rhythm. Under the atrium tracing shows a fast depolarization and a shorter action potential. Unlike the SAN, the atrium has a stable resting potential until it is stimulated. The atrial potential occurs just prior to the P wave on the ECG, showing atrial depolarization. Following the atrium is the AV node, shown here as the AVN. The tracing for the AVN is smaller and slower. The slope for the AVN is flatter, showing a delay in conduction, which allows the ventricles to fill completely before they contract. The Purkinje fibers show a rapid upstroke and a very long plateau. These fibers spread impulses rapidly throughout the ventricles. Following, we have the 3 ventricular muscle recordings, the epicardial, mid-myocardial, and endocardial. Each one of these has a plateau, but the durations are different. The timing differences contribute to the QRS complex and the T wave in the ECG. At the bottom of this figure, we have an ECG, beginning with the P wave, going into the QRS complex, and ending with the T wave. Starting here at the P wave is going to align with the atrial depolarization. The QRS complex is the rapid ventricular depolarization, and these points correspond to the steep voltage changes as the wave travels through the ventricles. Then we make it to the T wave. The T wave aligns the ventricular repolarization back towards the baseline. When looked at vertically, we can see each tissue's electrical behavior occurring in a sequence across approximately 200 millisecond intervals, ensuring filling and pumping. Here we are looking at the action potentials of atrial cells, Purkinje fibers, and ventricular myocyte comparisons. Although they have depolarization, plateau, and repolarization, their timing and shapes all differ. At the top of this figure, we have 3 different red action potential tracings. The atrial action potential on the top left, Purkinje fiber tracing in the middle, and the ventricular action potential here on the top right. The atrial action potential is going to be shorter and repolarizes more rapidly. Therefore, the cells are able to recover and become excitable sooner. The Purkinje fiber tracing shows a very fast upstroke and a long plateau because these cells have to conduct impulses quickly through the ventricles while maintaining proper timing. The ventricular action potential has a broader plateau phase, which prolongs the contraction time period. Under these three, we have a table listing the ion currents for each responsible phase. Ion A represents the fast sodium current producing the steep depolarization in contractile cells. ICAL, the L-type calcium current, which is number 2 here, maintains the plateau during phase 2. The funny current contributes primarily to pacemaker cells, but it does have a small part in supporting the conduction tissue. There are several potassium currents which repolarize. ITO creates brief notch in phase 1. IKR and IKS control a lot of the delayed rectifier phases, and IK1 establishes the resting membrane potential. Collaboratively, these currents shape how long or short the action potential is. On the right-hand side, we can also see the connections of currents to pharmacological medications. For example, the sodium channel blocking drugs like lidocaine and flectinide slow depolarization by reducing the ion A. The calcium channel blockers, such as verapamil and diltiazem, decrease ICAL entry, which shorten the plateau and reduce the contractility. Potassium channel acting medications like amiodarone, sotalol, zolpectamide prolong repolarization by reducing IKR and IKS, which increase the refractory period and help control re-entry arrhythmias. This figure as a whole allows us to see every arrhythmic drug class working by altering one or more of these currents. And if one change occurs, a single current can severely alter the cardiac action potential shape and or timing. Here we have the sarcomere. This is a structure which is the basic contractile unit of the cardiac muscle. The Z line are the boundary markers of each sarcomere. A bands contain thick myosin filaments. I bands contain actin filaments. The M line anchors the myosin filaments in the center. Tighten then stretches from the Z line to the myosin filaments, giving that elastic recoil, which helps maintain the alignment. According to Zedlak et al., the sarcomere is heavily linked to the electrical events of the action potential. During the plateau phase, calcium will enter through the L-type calcium channels and trigger additional calcium release from the sarcoplasmic reticulum. This is a process called calcium-induced calcium release. Calcium will then bind to troponin C, which shifts the regulatory proteins and exposes binding sites on the actin so myosin cross bridges can form. Zedlak and Collins emphasize that the smallest change in calcium entry significantly alter force production because contraction strength is directly tied to calcium availability. Titin also has a functional role by restoring preferred sarcomere length during relaxation, which helps maintain efficient contraction from beat to beat. Contraction occurs at the molecular level. At the top of the cross-bridge structure, we have thin filament, also known as actin. Along the actin strand, the troponin-tropomyosin regulatory complex is found, containing TNC, TNL, and TNT. During the resting state, tropomyosin sits over the binding sites on the actin, which prevent the myosin head from attaching. At the bottom of this diagram is the thick filament, which is the myosin backbone with the myosin head projecting out. The myosin head contains ATP binding sites and actin binding sites. There are two regulatory light change indicated here, one in yellow, the ELC, and one in blue, the RLC. These help stabilize and control the myosin head movement. The myosin protein C, located here in navy blue, helps organize the thick filament and fine-tune the contraction timing. When the action potential is reached by the cell, the calcium level will rise and bind to troponin C, which results in a conformational shift to move tropomyosin away from the actin bias site. This then exposes the actin binding site so the myosin head can then attach and form the cross bridge. ATP is hydrolyzed, allowing the myosin head to then pivot and pull the actin towards the center of the sarcomere to generate that force. ATP binding sites, again, allow the head to detach, and then the cycle can continue. The cross-bridge structure broken down at the molecular levels just shows how electrical events control the mechanical force by regulating calcium and give access to actin binding sites, giving the direct link to action potential directly to the contraction strength. Here we are looking at cross bridges switching between inactive phases and active states. On the left of the figure, the myosin heads are what we see in the off state. The heads are folded back towards the thick filament, backbone, and the two heads interact with each other. This head-to-head interaction prevents them from binding to actin. In the off state, the cross bridges are stored in reserve, which helps conserve energy when the heart does not need to generate strong contraction. On the right-hand side, over here, the myosin heads are what we call the on state. This is when the regulatory light chain, ELC in yellow and the RLC in blue, become phosphorylated, shown here with the arrow. Phosphorylation increases the flexibility and the rotation of the myosin head neck region, allowing the head to swing outward and become positioned closer to the thick filament. This movement increases the probability that the myosin head will attach to the actin when the calcium is exposed to the binding site. Czeslaw et al. Emphasizes this biochemical regulation works alongside the action potential. When calcium rises during the plateau phase, more binding sites on actin become available. At the same time, phosphorylation increases the number of heads that are ready to bind, allowing more force. Sherwood explains this mechanism allows the heart to quickly adjust contraction strength beat to beat without needing any changes in the muscle length. Prolonged action potential duration, as shown in the long QT syndrome, increases the risk of TDP, which is a polymorphic ventricular tachycardia, and sudden death. Medications like verapamil, diltiazem intentionally reduce the calcium entry and shorten the plateau, decreasing the contractibility, which is what we saw on the table a few slides ago. Antiarrhythmic medications alter sodium or potassium currents to stabilize rhythm. However, if these are misused, these medications can also cause arrhythmias. During ischemia, ATP depletion disrupts ion gradients, shortens the action potential, and leads to conduction disturbances. Understanding cardiac contractile cell action potentials allow physicians to properly interpret ECG changes and select appropriate medical therapies.